Lesson 13.3 on mutations. Our goal is to explain how mutations in the DNA sequence may or may not result in phenotypic changes, and also explain how mutations in gametes may result in phenotypic changes in offspring. Farsight says, "Here, Fifi, come on! Faster, Fifi!" Poor Fifi. All right, so our objectives are to define mutations and describe the different types of mutations, and also describe the effects mutations have on genes. Let's think about it. Two-head cat there. The sequence of bases in DNA are like the letters of a coded message. What would happen if a few of those letters changed accidentally, altering the message? What effects would you predict such changes to have on genes and the polypeptides for which they code? All right, so let's look at our first objective, define mutations and describe the different types of mutations. Now and then, cells do make mistakes in copying their own DNA, inserting the wrong base or even skipping a base as a strand is put together. These variations are called mutations, from the Latin word mutar, meaning to change. Mutations are heritable changes in genetic information. All mutations fall into two basic categories. Those that produce changes in a single gene are known as gene mutations, and those that produce changes in a whole chromosomes are known as chromosomal mutations. So, gene mutations first. Mutations that involve changes in one or a few nucleotides are known as point mutations because they occur at a single point in the DNA sequence. They generally occur during replication. If a gene in one cell is altered, the alteration can be passed on to every cell that develops from the original one. Point mutations include substitutions, insertions, and deletions. So under gene mutations, we have point mutations. Under point mutations, we have substitutions, insertions, and deletions. Let's take a look at each one of those. So substitutions. In a substitution, one base is changed to a different base. Substitutions usually affect no more than a single amino acid, and sometimes they have no effect at all. In this example, the base cytosine is replaced by the base thymine, resulting in a change in messenger RNA codon from CGU, arginine, to CAU, histidine. However, a change in the last base of the codon from CGU to CGA, for example, would still specify the amino acid arginine. The next type of point mutation, which is also again a gene mutation, is insertions and deletions. Insertions and deletions are point mutations in which one base is inserted or removed from the DNA sequence. If a nucleotide is added or deleted, the bases are still read in groups of 3, but now those groups are shifting in every codon that follows that mutation. Insertions and deletions are also called frame shift mutations because they shift the reading frame of the genetic message. Frame shift mutations can change amino acids that follow the point of the mutation and can alter the protein so much that then it is unable to perform its normal functions. All right, let's look at chromosomal mutations now. Chromosomal mutations involve changes in the number or structure of chromosomes. These mutations can change the location of genes on chromosomes and can even change the number of copies of some genes. There are 4 types of chromosomal mutations: deletion, duplication, inversion, and translocation. Okay, let's look at those first. Deletion. Deletion involves the loss or all of a part of a chromosome, as you can see from the example here. Duplication produces an extra copy of all or part of a chromosome. Inversion, ABC, ACB. Inversion reverses direction of parts of a chromosome. And translocation occurs when the part of one chromosome breaks off and attaches to another. So our objective there was to define mutation and describe the different types of mutations. Mutations are heritable changes in genetic information, and there are 2 types. There's gene mutations, which produce changes in a single gene, or chromosomal mutations, which produce changes in the whole chromosomes. And you are responsible for knowing each type of those gene mutations and chromosomal mutations too. So objective 2, describe the effects mutations can have on genes. All right, genetic material can be altered by natural events or by artificial means. The resulting mutations may or may not even affect an organism. Some mutations that affect individual organisms can also affect a species or even an entire ecosystem. Many mutations are produced by errors in genetic processes. For example, some point mutations are caused by errors during DNA replication. The cellular machinery that replicates DNA inserts an incorrect base roughly once in every 10 million bases. So small changes in genes can gradually accumulate over time. Stressful environmental conditions may cause some bacteria to increase mutation rates. This can actually be helpful to the organism since mutations may sometimes give such bacteria a new trait, such as the ability to consume a new food or source or to resist a poison in the environment. Mutagens. Some mutations arise from mutagens, which are chemical or physical agents in the environment. Chemical mutagens include certain pesticides, a few natural plant alkaloids, tobacco smoke, and environmental pollutants. Physical mutagens include some forms of electromagnetic radiation, such as X-rays and ultraviolet light. If these mutagens interact with DNA, they can produce mutations at a higher rate. Some compounds interfere with base pairing, increasing the error rate of DNA replication. Others weaken the DNA strand, causing breaks and inversions that produce chromosomal mutations. Cells can sometimes repair the damage, but when they cannot, the DNA-based sequence changes permanently. The effects of mutations on genes vary widely. Some have little or no effect, and some produce beneficial variations. Some negatively disrupt the gene function, though. Whether a mutation is negative or beneficial depends on how its DNA changes relative to the organism's situation. Mutations are often thought of as a negative because they disrupt the normal function of the genes. However, without mutations, as we'll learn about in evolution, organisms cannot evolve, because mutations are the source of genetic variability in a species. Some of the most harmful mutations are those that dramatically change protein structure or gene activity. The defective proteins produced by these mutations can disrupt normal biological activities and result in genetic disorders. Some cancers, for example, are the product of mutations that cause the uncontrolled growth of cells. Sickle cell disease is a disorder associated with changes in the shape of the red blood cells. Normal red blood cells are round. Sickle cells appear long and pointed. Sickle cell disease is caused by a point mutation in one of the polypeptides found in hemoglobin, the blood's principal oxygen-carrying protein. Among the symptoms of the disease are anemia, severe pain, frequent infections, and stunted growth. Some of the variation produced by mutations can be highly advantageous to an organism or species. Mutations often produce proteins with new or altered functions that can be useful to the organisms in different or changing environments. For example, mutations may have helped many insects resist chemical pesticides. Some mutations have enabled microorganisms to adapt to new chemicals in the environment. Plant and animal breeders often make use of good mutations. For example, when a complete set of chromosomes fails to separate during meiosis, the gametes that results may produce triploid, 3N, or tetraploid, 4N organisms. The condition in which an organism has an extra set of chromosomes is called polyploidy. Polyploidy plants are often larger and stronger than diploid plants. Important crop plants, including bananas and limes, have been produced this way. Polyploidy also occurs naturally in citrus plants, often through spontaneous mutations. So objective 2, describe the effects mutations can have on genes. Well, the effects of mutations on genes can vary very widely. Some have little or no effect, and some produce beneficial variations. Some negatively disrupt gene function. Mutations often produce proteins with new or altered functions that can be useful to organisms in different or changing environments. Our objectives were 1, to define mutations, describe the different types of mutations, and 2, describe the effects mutations can have on genes. Our goal was to explain how mutations in DNA sequence may or may not result in phenotypic changes, and explain how mutations in gametes may result in phenotypic changes in offspring. So both those objectives helped to support our goal. And here's another mutation picture for you. I don't know if it's real or not. Enjoy.